Everyone, and welcome to my YouTube channel Perfect Lab. In today's video we will learn about the Catalase test, which is a simple but important biochemical test which is used to identify bacteria that produce the enzyme catalase to catalyze hydrogen peroxide to water and oxygen gas. We will also cover the principle of the test, the procedure, result interpretation, and its significance in bacterial identification. So let's get started. Let's start our introduction by looking at the principles of the Catalase test. And before we look at the principles, what I want us to note is that the Catalase test is a rapid biochemical test which is often used to determine bacteria that have the ability to produce the enzyme catalase. And this test is a quick test which is being used to distinguish members of the genus Staphylococcus that are catalase positive from Streptococcus and Enterococcus species that are catalase negative. For the principle of the catalase test, the test is basically based on the ability of certain bacteria to produce the enzyme catalase, and this enzyme catalyzes the decomposition of hydrogen peroxide into water and oxygen, and as a result protect the cells from the toxic effects of hydrogen peroxide produced during aerobic metabolism. So now let's look at the positive reaction in the catalase test. So for the positive reaction, it means that the bacteria here have the ability to produce the enzyme catalase to decompose the hydrogen peroxide to water and oxygen. So when a drop of the catalase reagent, which is 3% hydrogen peroxide, is added to a bacterial colony that produces catalase, oxygen is rapidly released as visible bubbles to indicate a positive reaction or a positive test result as shown here. For a negative reaction, and what this means is that the bacterium or the bacterial colony here lack the ability to produce the enzyme catalase, and therefore will not be able to break down hydrogen peroxide into water and oxygen. So when a drop of the catalase reagent, which is 3% hydrogen peroxide, is added to the bacterial colony on the glass slide, there will be no production of oxygen, and therefore no bubbles will be observed as shown in the picture over here to indicate a negative reaction. So from the explanation, the catalase reagent or the hydrogen peroxide which is being used is serving as a substrate for the enzyme catalase produced from the bacteria to catalyze into the products water and oxygen. And the release of oxygen appears as bubbles to indicate a positive catalase test result. Let's look at the steps involved in performing the catalase test in the laboratory. The first step is to label your glass slide with your sample identification as shown here. The next step is to use a sterile dropper or pipette to place a drop or two of the catalase reagent, which is 3% hydrogen peroxide, onto the center of the slide. The next step is to use a sterile wooden applicator stick or a sterile plastic loop or a platinum loop to pick a small amount of a well-isolated bacterial colony from a fresh culture, which is 18 to 24 hours old. And once we are done with this, we mix the colony with the catalase reagent by emulsifying the bacterial colony in the drop of the hydrogen peroxide as shown here. So the next step is to observe for bubbles formation. Now, so let's look at how to interpret this result. So immediate bubbling after emulsifying the bacterial colony in the drop of the catalase reagent within 5 to 10 seconds shows a catalase positive result. whilst no bubbles formation shows a catalase negative result. And some key information that we have to note when performing these tests is that we are supposed to use fresh cultures and read results immediately, and we should also avoid colonies from blood agar since red blood cells contains catalase. Important note to consider when performing the catalase test. 1. Use fresh bacterial cultures, and these are cells grown in the laboratory within 18 to 24 hours for accurate results. Read the reaction immediately because delayed babbling may lead to incorrect interpretation. Do not use colonies taken from blood agar when possible. This is because red blood cells contains catalase and may produce false positive results. Use a wooden stick or platinum loop for transferring colonies, and try avoiding the use of iron-containing loops that may interfere with the reaction. And 5. Use only a small amount of inoculum, as heavy inoculation can make interpretation difficult. Quality control organisms, and these are known organisms that will either give a positive result for the catalase test or a negative result for the catalase test. So for the positive control, we will use Staphylococcus aureus as the known bacterium that will always give a positive result for the catalase test as shown here. And for the negative control, we will use the bacterium Streptococcus pyogenes, which will always give a negative result for the catalase test as shown here. So thanks for watching, and if you found this video helpful, please like, share, and subscribe to Perfect Lab. Leave your comments or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.